Okay, everybody, can we get started? Let's see. Okay, so I guess last time we stopped at this slide, right? Um, so this, I just wanted to go over again. This slide show us the, depend on the time period of the starvation, um, and of fat and starvation, and the whole period was divided into 5 different phases. Again, so the phase 1 basically is absorptive phase, like just a couple hours after meal and precisely uh, 3 hours, right, 2 to 3 hours after meal. So this time period considered the fat condition usually, right? That, and body basically utilize dietary glucose or exogenous energy source. Um, and then the phase 2, um, start from phase 2 is considered the post-absorptive phase. So phase 2 is pretty early starvation, right? Like 4 hours, after a meal. So phase 2 basically we able to use the glycogen because we have a glycogen, available. So majority of the glucose that we body utilize is from the glycogen breakdown, and particularly, in the liver. And liver glycogen also provide glucose to other tissues, right? For other tissues use, and muscle glycogen only for muscle itself use as energy. So and then the second phase, phase 3, start from 16 to I think about 20, more than 24 hours, uh, about 2 days, right? So this time period, because of glycogen, is almost completed. So body start to make glucose. So gluconeogenesis is the main source of the glucose for our body. So try to remember the time point, when glycogen is depleted. Um, 18 hours, right? Let's say 18 hours. So after 18 hours of starvation, and the glycogen is almost completely depleted. Um, and I also mentioned that, in addition to glucose, and because this diagram only talks about glucose utilization, but we know that we also utilize other alternative energy source, like fatty acids, ketone bodies. So after 1 day starvation, so the main energy source become different. I mean, the fatty acids have become the main sources of energy after 1 day starvation. And after 2 days of starvation, and ketone bodies also being made, right, from the liver. So ketone body and also become the important sources of energy, particularly for brain and skeletal muscle. And this table just show us the, different, again, similarly, different phases of the starvation and what sources of energy and used particularly for brain and other tissues as well. So again, phase 1, utilize exogenous or dietary carbohydrate is the main source of glucose for all the tissues. Of course, brain, right? So glucose is a major fuel of a brain. And phase 2, glycogen, particularly, especially hepatic glycogen or liver glycogen, and also gluconeogenesis also make glucose, right? But in phase 2, the liver gluconeogenesis is a major source of glucose, right? The kidney also make glucose, but during this second phase, the kidney contribute less glucose compared to the liver. So all the tissue except liver, muscle, and adipose tissue will use the glucose. So in other words, the liver, muscle, and adipose tissue use glucose, but at a small rate, right? Relative, the small rate compared to the other tissues. And brain still utilize the glucose as a major source of energy. And phase 3, glycogen, gluconeogenic become more important in terms of contribution to circulating glucose, right? Because glycogen start to be, depleted. So hepatic gluconeogenesis is a major source of glucose. And similarly to phase 2, most of the tissues still utilize the glucose, but the rate has slowed down, right? And brain is also, still use glucose as a major source of energy. And phase 4, the glycogen is completely depleted, and we don't have a glycogen. We use up glycogen, right? And gluconeogenesis becomes more important for the glucose as energy fuel. But meantime, the kidney start to become important sources of glucose, right? So kidney gluconeogenesis become important after 24 hours of starvation. And during this time period, glucose only use limited to some important tissues that heavily rely on glucose. For example, brain, red blood cells, right, and renal medulla, and some small amount of glucose can also be used by muscle. So during the phase 4, the body start to spare glucose, right, only for use by limited organs, tissues, and brain also start to use ketone bodies, but also try to spare glucose, but not enough glucose. So brain start to use ketone bodies, and if starvation continues, then ketone body will become most important energy fuels for the brain, particularly after 2 days of starvation, right? So just try to, understand this different time periods of the starvation and how body use a different, switch to using, different type of, different sources of energy fuels. Okay, now let's talk about the metabolic fuel utilization switch. So as I mentioned earlier, that the metabolic switch means that under fat condition, our body primarily uses exogenous fuel, right, dietary sources for energy. But when we start, starved, then we switch the utilization of energy fuel from exogenous to endogenous. And also another type of a switch that switch from utilizing carbohydrate in the fat condition to utilizing fat, right, fatty acids or ketone bodies during fasting. So two types of switch, metabolic switch, sources of switch, a type of energy fuel switch, right? So again, you probably try to remember the the timing, right? 18 hours I just mentioned. This time point is very important, determine whether glycogen is still available or depleted. So after 18 hours starvation, we, the glycogen is almost gone. So we cannot utilize the stored glycogen, stored glucose, and so we start to make glucose, right? So gluconeogenesis will become more active after 18 hours of starvation. And also the utilization of glucose will be slowed down as the starvation prolonged, right? Again, the purpose of this is try to spare glucose. And again, will not complete oxidized glucose, just partially oxidized glucose, right? To save lactate and a recycled lactate for making glucose. And brain, because of we mentioned, right, and red blood cells, these tissues are heavily rely on glucose. So brain still utilizes glucose just as a very late stage of starvation, and brain can start to use ketone bodies. And red blood cell always use the blood glucose, right? We always use the glucose as energy fuel. Cannot utilize the other energy fuels. And what happens after 24 hours starvation? As I mentioned, the gluconeogenesis, it then becomes the only sources of glucose after 24 hours starvation, right? And not only liver gluconeogenesis, the kidney gluconeogenesis also become important after 24 hours, right? So again, after 24 hours starvation, glycogen breakdown pathway being almost completely shut down because we don't have a glycogen anymore. So gluconeogenesis will become very active, and the two tissues, organs involved gluconeogenesis, kidney and liver, right? So after 24 hours, the kidney also make about 50% of glucose, contribute 50% glucose in there. And here's a question. So after 24 hours, and because the kidney become more important, so in terms of precursors for gluconeogenesis, amino acids, right, will be more important precursors for gluconeogenesis. And in addition to amino acids, some lactate, pyruvate, and glycerol were also, available for gluconeogenesis. And if you still remember, we mentioned that kidney mostly utilize which type of precursors for gluconeogenesis? Should you remember? Lactate, comparison lactate, amino acids, pyruvate, glycerol. Which type of precursor that kidney use most for gluconeogenesis? Amino acids, right? We talked about before. So amino acids is the most important precursors for gluconeogenesis in kidney. And here's a question: is acetyl-CoA able to make glucose? No. No, right? Acetyl-CoA is only has the 2 carbons, so acetyl-CoA is not able to be, used for to make making glucose. And other tissues, glycolysis activity is still high, and making lactate, right? And not going to complete oxidize glucose. So glucose is, can be converted from lactate. So again, after 24 hours starvation, most of the tissues utilize glucose and for glycolysis and release lactate, right? And lactate can be reused to synthesize glucose through gluconeogenesis. Remember the cycle, Cori cycle, right? So Cori cycle will be very active during the starvation. So brain is the only tissue that complete oxidize glucose still, under the very long fasting conditions. So we also have another question. So where do the amino acids for gluconeogenesis come from? We know that kidney utilize a lot of amino acids for gluconeogenesis. Where do these amino acids come from during the fasting conditions? Muscle, right? So muscle break down the proteins and release, fatty acids. And one of the cycle example, that amino acid can be used for gluconeogenesis. Remember the cycle? Alanine cycle, right? Alanine cycle. Okay. And more information about something happening after 24-hour starvation. And in addition to glucose, the fatty acids have become the major fuels for a lot of tissues, right? Particularly muscle. Muscle utilize lots of half of the fatty acids in circulation for energy. So again, after 24 hours of lipolysis in adipose tissue will be very active and then release fatty acids. So fatty acids level in the circulation should be high, increase right after 24-hour starvation. And most of the fatty acids will be uptake by muscle, half of, 50% of fatty acids picked up by muscle and utilize energy, and the rest of the fatty acids also can be picked up by other tissues and used for energy, except for one tissue of organs that cannot utilize, or actually two, at least two tissues that cannot utilize fatty acids. Which two tissues, organs? The brain, and what is the other one? We have a question in the test. Red blood cells, yes, right? So brain and red blood cells are not able to utilize fatty acids. And then the ketone bodies, right, will start increase, synthesized from the liver, right? And then the brain and muscle, again, are the two major tissues that utilize ketone bodies for energy, right? So again, brain cannot utilize fatty acids, and by able to utilize ketone bodies because of the red the ketone body much smaller molecules, right, be able to pass through, get into the brain. And kidney become the major and gluconeogenic organ, right? And liver gluconeogenic activities is decreased. So this probably related to these sources of precursors of gluconeogenesis, right? Because when starvation prolonged, that muscle break down proteins, and you have lots of amino acids available, right? So then kidney can use for gluconeogenesis. That probably is one of the reasons why kidney gluconeogenesis increase and liver gluconeogenesis decrease. And also the other reason is that because of the muscle, after 24-hour starvation, utilize more fatty acids and less glucose, right? So the lactate release will be also limited, after 24 hours. So you have again, probably have a reduced, availability of lactate during prolonged starvation. So that also is the reason for why liver gluconeogenesis activity is a little bit decreased, right? Because you have less lactate available. So. Okay, so this is the a diagram show you that the concentration of the different energy fuels during prolonged starvation. So as you can see very clear, the ketone bodies level significantly increase, right, and maintain a very high level. But glucose level actually significantly dropped, decreased, and amino acids, fatty acids, triglyceride level is also increased, and lactate glycerol levels actually decrease, right? So that's, I just explained, because why liver gluconeogenesis decrease and kidney gluconeogenesis increase, because of this, the precursors, the change during the prolonged starvation. Okay, so so again, the fatty acids, the ketone bodies, are the most important fuels, right, during the prolonged starvation. And glucose, ketone bodies, fatty acids, and triglycerides levels are increased during prolonged starvation, and glucose levels decrease, lactate levels decrease, amino acids levels decrease. So I just talked about the amino acids level actually increase. Why actually decrease? Why amino acid lactate decrease? In the circulation. In the circulation. Let's talk about amino acids levels, right, first. Anybody share idea? Yeah.
You say that the kidney uses amino acids for gluconeogenesis.
Right. Right. Yeah. So, muscle actually provide a lot of amino acids into circulation, but kidney picks up, right, and use a lot of amino acids for gluconeogenesis. So although muscle produces a lot, but kidney picks up, right? So, in general, net results, the consequences, right, it's actually amino acid level in the circulation relative low compared to, the fat condition. And why lactate levels decrease? The same reason, right? Firstly, because glycolysis, glucose decrease, particularly in the muscle. And secondly, lots of lactate being picked up, right, by the liver for gluconeogenesis. So in general, lactate level is also lower. Does that make sense? Right? Okay. Okay, now let's talk about Reynolds cycle. So this actually is a metabolic process that involves the competition of glucose and fatty acids for substrate utilization during starvation. And this theory is also applied to type 2 diabetes and insulin resistance condition. So that means the competition, that means if our body utilizes more glucose, then so some sort of mechanism can inhibit fat utilization. And vice versa, if we utilize more fatty acids, then it will inhibit glucose utilization. So this both energy fuel utilization kind of compete each other, right? So this series is actually named for Philip Randall, who described this theory in 1963. And it's just a simple diagram to show you the Randall cycle. For example, under the fat condition, we have a lot of carbohydrate available, so we utilize more glucose, right, and inhibits fat utilization. And during the fasting condition, because we have a limited carbohydrate, glucose available, and we utilize more fatty acids from the stored energy, right, from the fat, and inhibits utilization of glucose. This is a way of like a spare, glucose, right? I revised this slide a little bit. Just try to include, fatty acids. So that makes, I guess, probably will be better, more clear in terms of understanding. So Randall cycle, we also call it glucose fatty acid cycle, right? So glucose spared effect of fat-derived fuels or fatty acids. So fatty acids has a glucose spared effect, right? So mean that if we utilize more glucose, we inhibit our body to utilize glucose, right, to spare glucose utilization. And so if you look at under these two different physiological states, for example, in the fat condition, right, we have a high blood glucose, right? Glucose basically is from diet. And this will increase the insulin secretion and decrease glucagon. So the insulin to glucagon ratio is increased, right, during the fat condition because of a high blood glucose. Stimulate beta cell, right, a pancreas to secrete insulin. And what happens, the change, insulin to glucagon ratio is, insulin we know is always promote the anabolic metabolism, right? So promote the energy storage for both glycogen and also fatty acids. So fatty acids storage being promoted because we have a high levels of insulin and low levels of glucagon. And the mobilization of fatty acids will be inhibited, right? Again, also because of high insulin and low glucagon. So what is consequences is that our body stored more fatty acids and inhibits the fat utilization, right, or fat mobilization. So that means we have less fatty acids available in the circulation, right? So that will increase our body to utilize more glucose as energy. It's just a simple, process, the simple explanation, right, for this process. And during the fasting condition, because our blood glucose levels drop and insulin secretion decrease, but glucagon secretion increase, right? So the ratio of insulin to glucagons decrease. So the effect of this change of hormone to the fat storage and fat mobilization is just opposite, right? Low insulin levels will stimulate lipolysis, stimulate fat mobilization, right, from fat tissue, and inhibits the fat storage. So that means we have more fatty acids that being released from fat tissue, and we have more fatty acids available. And so we can use fatty acids. I do not need to use much more glucose for energy. So glucose utilization will be decreased or inhibited. So this is just, again, the simple model, how the Reynolds cycle or glucose fat cycle works. Any question about this Reynolds cycle? Okay. And this just put the tissue. It's a similar, model, but just that it's plotted a different way. That involves two important tissues, muscle and adipose tissue. Adipose tissue, why is it important? Because it's the major tissue that stores fatty acids, right? And muscle is the most important tissue that utilize fatty acids. So when we have, for example, in the fat condition, when we have a lot of fatty glucose available, and fat tissue, adipose tissue, will utilize glucose, and this will inhibit the breakdown of triglyceride, right? So we have less fatty acids available. And under the fasting condition, because we have lots of fatty acids being available from fat tissue, so in the muscle, we utilize lots of fatty acids for energy, and this will inhibit glucose utilization, so spare glucose during the fasting conditions. Right? And this is actually the next two slides is talk about the mechanism, and just give you detailed understanding how this Reynolds cycle works, right? And this is the slide represent the physiological condition under fasting condition, right? So the blue area actually represent circulation, right? The blood, and this the whole thing is a cell, and this is in the cytosol, the yellow, right, the dark yellow area represents the cytosol, and light green area is the mitochondria, right? So under the fasting condition, we have high levels of fatty acids, right, that basically released from fat tissue. So cells, I believe this is muscle cells, right? So cells and takes up fatty acids from the circulation and utilizes for energy. So if you still remember that CPT1, right, the key transporters, and that located on the mitochondrial membrane that can transport fatty acids from cytosol into mitochondria for beta oxidation and generating acetyl-CoA, right? If we have lots of fatty acids available, and we can utilize a lot of fatty acids, oxidize it, and generate lots of acetyl-CoA, right? So acetyl-CoA level will be increased during fasting condition when we increase fat oxidation. And acetyl-CoA is important substrate that can block or inhibit pyruvate dehydrogenase enzyme. If you still remember, right? This is the inhibitors of pyruvate dehydrogenase enzyme, and then can block the glucose oxidation pathway. So this is a glucose oxidation pathway, right? Glucose, if you remember, uptake by GLUT4, and the first reaction enzyme is hexokinase in the muscle, right? Hexokinase and oxidize the metabolized glucose, and this is a glycolysis pathway, right? To pyruvate enter the mitochondria for further oxidation. So if this is a key point, a key reaction, right, for glucose oxidation. So if this reaction is blocked, so glucose cannot be further oxidized to acetyl-CoA and complete oxidation, right? So then can stop, glucose oxidation only at the glycolysis pathway, right, at the pyruvate. So pyruvate may be eventually be converted back to the lactate, right? And this is the one important mechanism, right? How increased fatty acid oxidation or decrease glucose oxidation by increasing acetyl-CoA and inhibits pyruvate dehydrogenase. And it's also the second mechanism is that because you have a high levels of acetyl-CoA can be converted to citrate, right? So citrate be transported back to the cytosol and converted back to acetyl-CoA and for malonyl-CoA formation. And this is the pathway for lipogenesis, if you still remember, right? So this is a key reaction in the lipogenesis and catalyzed by the key enzyme. Can you guys tell me the key enzyme for this reaction? For the formation of malonyl-CoA from acetyl-CoA? Yeah.
Is it the HMG-CoA reductase?
No. Yeah.
ACC?
Yes, ACC, acetyl-CoA carboxylase, right? So ACC is a key enzyme in the lipogenesis pathway. And there is another enzyme, and I don't think I asked you guys to remember, right? But ATP citrulase. So this enzyme is the breakdown citrate to acetyl-CoA. And this enzyme, ATP citrulase enzyme, activity usually highly regulated by insulin. When insulin level is high in the fat condition, then ATP citrulase activity decrease. So that would causing the accumulation of citrate. So citrate cannot be, break down to acetyl-CoA for malonyl-CoA production. So citrate level will be increased and malonyl-CoA level will be decreased, right? So and citrate is the inhibitors of these two key enzymes in glycolysis pathway, right? So yes.
So on this diagram, you're saying that fatty acid utilization is inhibiting the glucose utilization.
Yeah, right.
And you have it written that citrate increases.
Mm-hmm.
And then you were saying that citrate gets converted to acetyl-CoA and then malonyl-CoA and fatty acid oxidation happens. Kind of like that whole process, but it also says that it's going down. You're just saying that because of insulin as an inhibitor?
Yeah, this one.
But would insulin be active in this diagram technically?
No, sorry. Insulin stimulates this, but during the fasting, insulin levels decrease, right? So this enzyme will be also decreased because of lower insulin level during the fasting, right?
So would malonyl-CoA be up actually in this picture? Production of malonyl-CoA?
No, malonyl-CoA actually decrease, right? Because this enzymes decrease, right? So the conversion of citrate to acetyl-CoA will be also lower
What causes ATP citrate-lyase to go down? What inhibits
because of lower insulin levels,
because of low insulin?
right?
I thought you said because of high insulin on the side.
Well, I mean, the insulin basically stimulates this enzyme activity, but that during the fasting condition, we have a lower insulin, right? So this enzyme activity is also decreased because of a lower insulin level during the fasting condition, all right? So all this pathway actually slow down or decrease. So you have a less malonyl-CoA that's produced, but you have a more citrate accumulated. And citrate, again, is the inhibitors of these two key enzymes in glycolysis, right? So try to remember under the fasting condition, the reason why utilization of fatty acids, increased utilization of fatty acids will inhibit glucose utilization. Two mechanisms, right? One is because of beta oxidation, increase generate more acetyl-CoA. So acetyl-CoA inhibits pyruvate dehydrogenase activity. So we'll block pyruvate further breakdown to acetyl-CoA, right, for further, oxidation. And the second mechanism, because of citrate levels increase, but you just understand why citrate levels increase, but you probably don't need to remember, ATP citrate lyase, right? So second mechanism is citrate levels increase, right? So citrate is an inhibitor of two key enzymes in glycolysis pathway, right? So the whole pathway of glucose utilization will be inhibited during the fasting condition because, due to the increased fatty acid oxidation, right? Any questions? Okay. Yeah.
So citrate levels increase because of the ATP citrate lyase.
Right, citrate lyase activity decrease. Yeah. Yeah. Okay. All right. So under the fat condition, we have glucose available, right? And we utilize glucose, and so this pathway will be increased. And what happens that the citrate convert to acetyl-CoA and malonyl-CoA production pathways also increase. As I mentioned, the citrate lyase activities increase, right, because of high levels of insulin in the fat condition. So ATP citrate lyase activity increase, so can convert it more citrate to acetyl-CoA, and ACC level is actually also increased, remember, by insulin, right? So both enzyme, ATP citrate lyase and ACC, both enzyme activities are increased because of high levels of insulin in the fat condition. So that will make this pathway more active to produce more malonyl-CoA. So malonyl-CoA, do you guys still remember how malonyl-CoA regulates CPT1? Inhibits, right? Yeah, inhibits. So citrate, malonyl-CoA inhibits basically CPT1. So then blocks fatty acids transported from cytosol to mitochondria and block beta oxidation. So this is relatively simple, right? You just need to remember during the fat condition, because of malonyl-CoA production is increased, so cell has high levels of malonyl-CoA, which can inhibit CPT1 and inhibits fatty acid transporter and beta oxidation. Right? Any questions? Do we need a break? Maybe 5 minutes break, right?
Quiz at the end of class or
Oh, you haven't done it? So yeah, sure.
okay.
You want to do it end of the class?
I could do it in class or I could do it whenever works best.
Okay. Um, yeah, maybe end of the class. Is
End of
that
class?
okay?
Okay. Thank
Yeah.
you.
Yeah. Welcome. Mhm.
Professor,
Yeah.
I didn't get this one. Los
Mhm.
muscle glycogen contribute to blood glucose and why? And I didn't.
Oh, because of the enzyme, uh, glucose 6 phosphatase.
So it does contribute.
Yeah, yeah. Muscle lacks of this enzyme, right? So it can
Lactate and the enzyme, depending on
lacks. I mean, does not muscle does not have a glucose 6 phosphatase,
Oh yeah, that's true.
right? So
Yeah.
that the glycogen breakdown will not be complete. Finally release the glucose, but we just utilize in within the muscle.
Okay.
Yeah, that makes sense.
Yeah.
Okay.
Thank you.
Yeah. All right. let's see. Okay, now let's start talk about the regulation of fuel utilization, right? See, just help us understand why, when we utilize different sources and different type of the fuels during fasting condition, right? And we, switch the fuel utilization. The hormone actually is very important factors that involve this regulation, right? And enzymes, of course, is the target of the hormones, right? Hormones basically regulates enzyme in different pathways and regulates, metabolism, fuel utilization. And lastly, the transporters, right, like group 4, is also the key that can control the flux of metabolic fuels into the cells, right? So 3 different levels regulation: hormonal regulation, which is most important, and the second level is the enzyme, right? That pretty specific in the metabolic pathway, and lastly the transporters also that involve in the flux of the metabolic fuels into the cells. And this, I think we talked about before, and about how our body maintain blood glucose level, and 3 main major organs, tissues that involve in this regulation, called glucose homeostasis, right? Pancreas, liver, and 4 actually. Pancreas, liver, muscle, and adipose tissue, right? So these 4 major metabolic tissues. And one tissue is basically secrete insulin, right? The pancreas, and the other 3 tissues, the major insulin-sensitive tissues. Okay? And we just talk about individual, right? The different states. First, in the fat condition, when we ingest the carbohydrate, we raise our blood glucose level, right? And this will lead to first here response. You guys still remember, right? High blood glucose levels stimulates insulin secretion from pancreas, right? So insulin levels decrease and glucagon levels decrease. So which cells in the pancreas that secrete glucagon still? Do you guys know? Beta cells secrete insulin. Alpha cell, right? So alpha cell in the pancreas secrete glucagon. So glucose stimulates insulin secretion from beta cell, but inhibits glucagon secretion from alpha cell. So the ratio of insulin to glucagon is increased, right? High insulin, lower glucagon in the fat condition. And this will then affect 3 organs, tissues. First, in the liver, insulin will promote glycogen store, right? So convert the glucose, carbohydrate, stored carbohydrate glucose in the glycogen liver, and also converted glucose to fat, so to stimulate lipogenesis. But insulin can also does something that inhibits glucose production from the liver. So glucose production, actually 2 metabolic pathways contribute to glucose production from the liver. Can you tell me the 2 glucose metabolic pathways that can contribute glucose production from the liver? Gluconeogenesis, yes, and when the very early stage of starvation, glycogen, right? Glycogen breakdown. So in a fat condition, we don't want it to break down glycogen. We don't want the glucose, produced. So insulin, at meantime, also inhibits glycogen breakdown and inhibits gluconeogenesis. So all this affect the metabolic activity together will decrease glucose production from the liver, right? And muscle, during the fat condition, we have high levels of insulin, then transporters that are in the muscle, which one? Group 4, right? Group 4 will be very active, can be stimulated by insulin, then will take up lots of glucose into the muscle. So muscle is a very important tissue that in terms of the role in the rolling blood glucose, right? So muscle takes a lot of blood glucose from the blood and reduce the blood glucose level. And adipose tissue is also the tissue that can uptake glucose from the blood, also by group 4, right? So all these 4 organs work together to reduce blood glucose level, to not allow blood glucose level to be too high under fat condition, right? And during the starvation, again, all these 4 organs work together, right? In the pancreas, during the starvation, the alpha cells secrete more glucagon, and beta cell secretes less insulin. So glucagon levels increase and insulin levels decrease under the starvation condition. And again, this will also affect the 3 tissues, right? Liver, of course, during fasting, glucose production will be increased, right? Increased glycogen breakdown and increased gluconeogenesis will allow more glucose secrete from liver and to raise the blood glucose level. And muscle, under the fasting condition, we don't want the muscle to take up too much glucose, right? That will causing more decreased blood glucose level. So we try to inhibit group 4 translocation, inhibits glucose uptake by the muscle and also by the adipose tissue, right? So all again, these organs coordinately regulates blood glucose level and try to maintain glucose at the normal level, at the normal range, right? And here I just wanted to add glucose homeostasis under the exercise condition. So we mentioned earlier the catecholamine, like epinephrine, norepinephrine, right? Also it regulates blood glucose level, and this usually is increase catecholamine levels during exercise and also the stress. So the liver, muscle, and adipose tissue are the main targeted tissue of the catecholamine. And so in adipose tissue, epinephrine can increase the lipolysis, right? So increase, the fat release, the fatty acids release. And in the muscle, catecholamine can increase the glycogen breakdown, right? And also amino acid catabolism. And in liver, increase gluconeogenesis. So again, in terms of a glucose, regulation, the the main effect of the catecholamine is to regulate gluconeogenesis in the liver, right? So during exercise, because lots of glucose is being taken up by the muscle for use for energy, for ATP production, and so body also try to make more glucose. So liver is a main target of epinephrine during the exercise, in terms of blood glucose level maintains. Okay. And here on this slide, I just want you guys to talk to each other. Right? This just show us how liver plays a role in the glucose homeostasis, right, during the fasting or fat condition. Right? So take a couple minutes and talk about it.
Okay.
All right. So, can you guys talk about it in the fat condition, how liver functioning and in maintaining the blood glucose level in the fat condition? Which metabolic pathways? Let's talk about the hormonal level first, right? In a fat condition, what is the ratio of insulin to glucagon? High insulin, low glucagon, right? And what about the all the glucose metabolic pathway activities in the liver? So under the fat condition, we have higher insulin level, low glucagon, right? And we have, the blood glucose level will be high, right? And our body try to reduce the blood glucose level, right? So how liver, make contribution to, so, sorry? Stored, yes. So the stored uptake glucose and stored in the glycogen, right? So glycogen synthesis pathway should be high. And then, and what about glycogen breakdown pathway? Should be lower, right? We don't want to break down glycogen. And gluconeogenesis also lower, right? So we have a lower glycogen breakdown, lower gluconeogenesis, and high glycogen synthesis, right? So all this work together, and we have less glucose produced from the liver, right? We want to inhibit glucose production from the liver and the fat condition. And how about the fasting condition? We have a hormonal levels, insulin lower, right? Glucagon high. And what we need to do, we have a lower blood glucose level, right? So we wanted to raise blood glucose level. So what is the contribution of the liver? Yeah, break down glycogen, release glucose, right? And also gluconeogenesis, right? Making more glucose. And the synthesis will also need to be, we would inhibit, right? Inhibits glycogen synthesis. So again, glucose production from the liver increases, right, during the fasting condition, right? Okay, here's just more information about, the insulin and the glucagon, how they regulate metabolism. They actually does the opposite, right? Regulate the effect. So metabolic pathway, insulin basically is the anabolic hormone. That means that usually stimulates synthesis, right? It stimulates glycogen synthesis, stimulates triglyceride synthesis, and stimulates the lipogenesis, fatty acid synthesis, right? And decrease the, the catabolic pathways, right? It decrease glycogen breakdown, decrease gluconeogenesis, and decrease lipolysis, right? So here I just put all the, metabolic pathways of glucose and lipids together, and protein, right? So insulin stimulates protein synthesis. So in general, it's a anabolic hormone. So it should be easy to understand, to remember, right? And in terms of metabolic enzymes, that in the liver, the glucoutilization and glycogen glucokinase it should be increased, right? So increase glucoutilization and decrease gluconeogenic path, key enzyme like PEPCK and glucose-6-phosphatase, right? And glucagon does just the opposite effect of insulin, and basically like promotes the utilization of stored energy, the breakdown, triglyceride breakdown, the glycogen. So we have two tables that I don't want to go through, but you guys can wanted to go through and see if you understand all this regulation at the three different levels, hormonal levels, enzymes, key enzyme in each pathways, and also transporters, right? Okay, now let's talk about diabetes. So hopefully something more interesting. So we have two different types of diabetes, right? Type 1 and type 2. So type 1 diabetes, what is the cause of type 1 diabetes, you guys know? And no, and beta cell deficiency, right? So then beta cell are not able to make insulin. So have no insulin or little insulin in the type 2 diabetic patients. So the treatment is relatively simple. Just give insulin supplementation. The issue is that the way to give insulin is kind of a challenge, right? You have to make sure that the timings when you need insulin and the dose is kind of very difficult to control, I guess. And also the route for the insulin administration, the injection is pretty challenging. And type 2 diabetes. So the cause of type 2 diabetes is insulin resistance. So it means that particularly at the early stage of the disease, and patients is able to make even more insulin, but insulin action is reduced, so insulin cannot efficiently function, right? So they call it insulin resistance. But under the later stage of type 2 diabetes, then somehow beta cell also have problems, not able to make insulin. So only at very later stage of type 2 diabetes also need insulin for treatment. So treatment is it's kind of complicated, and we do have some anti-diabetic drug available to control blood glucose level, but I don't think the effect is kind of optimal. So usually you need to have a different treatment combination of the treatment. For example, you try to change the diet, right? And try to exercise, and combine with diabetic drug, and also later stage you need insulin therapy. Here I just wanted to talk about the mechanism, right, to understand better the disease. And this is actually complicated metabolic disease involved in multiple tissues. And here I just wanted to talk three major tissues: adipose tissue, muscle, and liver, and see what is the problem in these different tissues in the type 2 diabetes. So first is adipose tissue. So the main problem in adipose tissue in type 2 diabetic patients is the GLUT4. GLUT4 expression level actually decreased in the type 2 diabetes. And not just the levels decreased, but also translocation, from intracellular to cell surface. This process is also has a problem. It cannot efficiently transport GLUT4 to the cell surface. So that causing problem, right? To uptake glucose from the circulation. And here's just the pathway that you can see. The insulin is just represent the part of the cells, right? Fat cells, and on the fat cell surface there is insulin receptors, and also GLUT4, right, to uptake glucose. And this part of it just represent kind of plasma, circulation. So insulin binds its receptor on the cell surface and transduces signaling to induce GLUT4 translocation to the cell surface, and this process is reduced in the in the type 2 diabetes in adipose cells because of insulin action is reduced, right? So insulin cannot efficiently induce GLUT4 translocation to the cell surface, and this will lead to decreased uptake of glucose, right? So, and you have less glucose in the cells and more glucose remain in the circulation. And in addition to the glucose metabolism, type 2 diabetic patients also have a problem with lipid metabolism, right? So insulin and also has effect on the lipid metabolism. For example, the HSL is a key enzyme, right, for the triglyceride breakdown or lipolysis. And in the type 2 diabetic patients, and because of insulin action is reduced, so insulin cannot efficiently inhibit HSL, so that will lead to increased HSL activities, and this will causing increase the lipolysis, right? Increase both fatty acids release into the circulation, and also LPL, lipoprotein lipase. This enzyme activity we know is also regulated by insulin, right? So insulin resistant in diabetic patients, the LPL cannot be efficiently produced and released and to hydrolyze triglyceride in the circulation. So this will causing the more triglyceride accumulate in the circulation, right? Does that make sense? So all this change that related to insulin resistance, and because of fatty acids level is high in circulation, so ketone bodies will also be high, right? If you still remember the regulation of ketogenesis, one of the important factors is the high levels of fatty acids, right? So in type 2 diabetic patients, they usually have high blood glucose level, high fatty acids, and high triglyceride level, and also high ketone body, right? So if you probably know that type 2 diabetic patients usually have a kind of, special kind of smell, right? The breath, because of the high levels of ketone bodies. Okay, now let's look at the muscle. So what's wrong in the muscle in type 2 diabetic patient? So GLUT4 expression level basically is normal, right? The GLUT4 level is okay, but translocation of the GLUT4 to cell surface is decreased. So this will cause the same problem, right? You have a glucose uptake significant decrease, so that is a big problem, right? We know skeletal muscle takes a lot of glucose, so if this cannot take that much glucose, and will causing high levels of glucose. And muscle is also important tissue that oxidize fatty acids, right? In the type 2 diabetic patient, because of lipolysis increase, have a high fatty acids levels, and plus muscle cannot efficiently oxidize fatty acids, so that makes things worse, right? So we have an even higher level of fatty acids in the circulation. And the liver. And just thinking about what insulin does in the liver in terms of glucose metabolism, so it could be also easier to understand, right? Liver insulin actions decrease, then you have a decrease, the glycogen synthesis, and you have increased glycogen breakdown, and also increase gluconeogenesis, right? So in general, the type 2 diabetic patient liver makes more glucose and secrete more glucose into circulation, so it makes things worse, right? So together, uptake glucose by muscle and adipose tissue decrease, and increase glucose production from the liver. So these two things come together, and makes glucose levels very high. And also in type 2 diabetic patient, the fatty liver disease is very common. The reason because they have a high levels of fatty acids, right? The fatty acids will pick up into the liver and makes triglyceride and accumulate in the liver. Okay, I believe this is the last slide. So insulin resistance, usually people think they have a metabolic inflexibility. What does that mean? And let's go to the details. So for example, in the lean, the normal insulin sensitive subject people, under the fat condition, when we have a high glucose, a carbohydrate available, and we were able to stimulate insulin secretion, right? And insulin should be efficiently act to increase glucose utilization, to decrease gluconeogenesis, decrease liver glucose production, so then be able to maintain blood glucose well. But in the obesity or insulin resistant conditions, particularly when we eat a meal, we're causing the blood glucose level, but body cannot handle this, cannot efficiently utilize uptake and utilize glucose, and cannot suppress gluconeogenesis, right, and suppress glycogen breakdown. So then we can increase the blood glucose levels. So this sort of kind of metabolic inflexibility. That means we diabetic patients handling glucose, inability to handle glucose has a problem, right? All right. Any questions? No? Okay. Any other question for exam or stuff? Oh, okay. Let's see. So we have a couple of summary slides. Yeah, I guess we'll just stop here for today, maybe a little bit early.